So good afternoon, everybody, and welcome to the first webinar of the VALITEST webinar series on the use and validation of high-throughput sequencing tests for diagnostics in plant health. For the one that are joining a VALITEST webinar for the first time, so VALITEST is an EU-funded project undertaken by a consortium of 16 partners and built to improve the reliability of diagnostic tests performed in plant health laboratories across the European and Mediterranean region. The project has three main objectives. So the first one is to provide a more complete and precise description of the performance of diagnostic tests. The second one is to stimulate, optimize, and strengthen the interaction between stakeholders in plant health for better diagnostic. And the third one is to lay the foundations for structuring the quality and commercial offers for plant health diagnostic tools. So you can find more information on the project on our website. So this series is about high-throughput sequencing, which is a powerful tool that enables to simultaneously sequencing of potentially all organisms present in a sample. And because of the growing interest in applying HTS for routine diagnostic in plant health laboratories, the objective of this webinar series and activity is to present the recommendations that were developed by the work package 2 of the VALITEST project on the process of selection, development, validation, and verification of high-throughput sequencing tests, and the quality assurance of their routine use as diagnostic tests. So these recommendations are general and to enable a broad application in all plant health fields and with appropriate flexibility to account for the changes of technologies. So today's webinar will be an introductory session and will give you an overview of the HTS process, including the different steps in the laboratory and for the bioinformatics analysis. So the presentation will be recorded and a link to the recording and to the PDF presentation will be provided on the VALITEST website. And just for you to know, some polls will be asked to you, but not directly in the GoToWebinar platform, but in WooClap. And so you will need either use a mobile phone or a web navigator to answer them, and we will give you further information how to do that later. So our speaker today is Sébastien Massart. Sébastien is a plant pathologist interested in diagnostics improvement, plant virology, and plant microbiome research. He is currently leading a research of 10 scientists in plant pathology. And Sébastien is also a professor at the University of Liege in Belgium. So with no further ado, I will give the floor to Sébastien for this first introductory session of the series.
Okay, thank you very much, Charlotte. So this is, as mentioned by Charlotte, this is today an introduction to high-throughput sequencing. If you want to go further, there is the webinar 2 and 3, which are already, which are still available and open for registration, which will go a bit more in details on how to prepare the laboratory for conducting high-throughput sequencing tests. And the third one on Tuesday, this is how to develop, validate, and routinely use the high-throughput sequencing for diagnostic purpose. So today, big question. I will try to share with you information in order to respond to a single question, in fact. What is high-throughput sequencing? So for that, and before going into the high-throughput sequencing itself, let's make a bit of history from where we come and allows us to understand better where we are and how we can use the harvest the technological evolution. Here is like prehistorical painting representing the sequencing in the 90s. We were able at that time to sequence somehow like 1,000 nucleotides a day. For those who might remember, this was like this polyacrylamide gel with radioactivity. I did some of them. It was really a complex, lengthy process. So having some sequence was really not so easy. Later on in the year 2000, the end of the 90s decade, there was the first industrialization, let's say. I can see here in the modern time of Charlie Chaplin, it's like sequencing start a big development, and we were able at that time to sequence 1 million base a day, already a nice improvement. And that was the time of starting the sequencing of the human genome, which was published in the early year 2000. And this was like a big project with hundreds of scientists and billion of euro of budget, mainly carried out in the United States. So this project allows also to progressively improve the throughput of high throughput sequencing and correspondingly to decrease the cost. During the first phase of the year 2000, the evolution was like this old car. It was evolving quite significantly, but this was not yet the booming of high throughput sequencing. The real thing is this evolution, the throughput and the cost evolution followed the one of the computer. You know, the computer, every 18 months, for the same price, you get the double capacity, and it's true from the year 60s. And this is, sequencing was following this trend in terms of throughput and price dropping. The big evolution with this bright, rapid car came into the middle of the year 2000 with the advent of high throughput sequencing, which was first used mainly for human and even human pathogen, but progressively a bit more than a decade ago, it started to be used for plant pathogen or plant pests. And today, last year, in fact, but a few months ago, we were able to look at this. It's about 100,000 billion base a day for which we were able to sequence. That's a huge difference. If you look, we started from 1,000 per day to 100,000 billion a day. So no other technology has come so fast in this time frame, with a clear exponential acceleration in the last decade. So it raised interest in the diagnostic of plant pests, whatever they are. A lot of work has been on viruses, but it's also very interesting for bacteria, fungi, nematodes, insects, or other arthropods, or even invasive weeds. There are applications of high throughput sequencing to scan the pollen, for example, of plants, including even invasive plants. So this is therefore an exponential curve of high throughput sequencing. Exponential curve in terms of output and throughput per day, but also a reduction in the cost, making it more and more affordable for plant pest diagnostic. And I wanted to share with you something with the exponential points. Here is a chess square with 64 cases. And I would like to tell you a story. This is a story of the Sultan of Sissa, somewhere in Asia. And this Sultan was very, very rich. One of the richer sultana in Asia. He does everything that he wants, but he gets sick. And nobody was able to cure him. So a foreign physician with a lot of knowledge into how to cure people was traveling over there, and the Sultan asked him to go to him and say, hey, guy, I know you have a very, very good knowledge to cure all the people, any kind of disease and whatsoever. Please, I have something, nobody can cure it, help me. And the doctor say, okay, but what's the price? Ask me something, I will think about it, but I'm very rich, so if you cure me, I can make you rich also. And the doctor just say, you see the case here, A1. I will put a single grain of rice, a single one. And what I just asked you, in fact, is that on the case A2 you put 2, there you put 4, 8, and so on. And you just fill this wool table. The Sultan was laughing. Hey, okay, guy, I will give you a few well, I mean, a few kilograms of rice. That's okay. That's okay for me. We have to deal. And no, the doctor, which was actually very good, he cured the guy. At the end of the story, what was the cost for the Sultan? So for that, we will make a first trial of how it works, the interaction. So if you have a mobile phone, please just scan it, this QR code. If you don't have a mobile phone, just use this address, so www.wooclap.com/OCIPBU. Let it on the screen, and you will land to a page. You will not be able still to participate. You will have a message stating that you should wait. So I let people going there. Have in mind, once we are there, stay there. Let it open. We will use it throughout the training today. So I let you arriving progressively over there. I show it in another way here with the web page. So here again, you have the web page here. So let's make a poll together. I ask you to all of you, what is for you the quantity of rice given by the Sultan? And I will just hide the response. We just answer what you feel, what you know. We have already 6 responses. So let's go. It's a guess. If you are strong in mathematics, it can help you. Let's go. We have like 25 votes. Again, you can scan the QR code here. It's fully anonymous, so even if you say something wrong, nobody will know. So use it. Okay, I will just show you what's the current pool right now. And that fact, we have mathematicians also among the participants, because in fact, most of you the majority of you gave the good answer. In fact, how many grains of rice should give the Sultan to the doctor? It was 2 exponent 64, which means that's actual very big number. And this number, to have it in mind, it corresponds to 1,000 years of the current production of rice. And this is important to have it in mind because, in fact, that means that the exponential, most of the people don't have in mind the importance of the exponential and how it can change the game. And that's the point here with high throughput sequencing. This exponential evolution changed the game, and change the game also will continue to impact the research, but more and more know the diagnostic too. So let's go further and let's have a look on the steps. That's, after this very first introduction, the steps of high throughput sequencing once you want to carry out. There are one part, sampling. I mean, this is needed for any test you want to make. The blue parts here that you can see from 2 to 4 are the parts linked to the laboratory. What are the steps that have to be carried out in the laboratory? Nucleic acid extraction, the library preparation with 2 modalities, the sequencing itself. Once you generate the millions of sequences, there is a big part which is needed. This is the bioinformatics. And for bioinformatics, there are 3 main steps. Whatever the protocol that you are using, there is first an analysis of the raw reads. We will analyze what has been generated by the sequencer, check the quality, eliminate sequences that are not interesting or that can create bias once you make the analysis. After that, there is the identification of the targets, and we deliberately state targets because it's not only pests that can be detected by high throughput sequencing, and we will also make analysis of controls. And that's something which we'll exchange with you during the webinar on Tuesday, the importance of the control and how to change our mind related to the control of high throughput sequencing. And at the end, again, for the diagnostician, the confirmation of the result, their interpretation, and how to report them. In fact, all of this is like the backbone of the guidelines that we have generated in the frame of VALITEST. I will discuss, I will show you a little bit more on the guidelines also Tuesday. And today, the objective is to review these different steps, to introduce you to the different steps, so you have in mind what does it correspond, in fact, these steps. So let's go first on the laboratory protocols. So laboratory protocols, it starts by sampling. And sampling, I mean, there is no difference with any other test. You have to take a sample that it is representative of the plants, of the field that you are, of the batch that you are sampling. So basically, you will apply similar rule as needed for any other techniques. They might be minor adaptation, but at the end, it's the same. You need to have a sample which is representative of the what needs to be tested. And so, same problems as any other test, no really novelties. And we have to keep in mind that the matrix that we will use can be either and will be a complex of multiple organisms. Even if you take plants, these plants host many fungi, bacteria that are present within the tissue. You can also, as a sample of having an environmental sample, soil, water, whatever air. You can start apply high throughput sequencing from spore trap or from insect trap. And that's application that are currently developed. You use a spore trap and you apply high throughput sequencing to determine which fungi are present there. You can do the same with insects. Large scale trapping of insects, and just you extract the DNA from the wool composition and you sequence it. So once you apply high throughput sequencing, it's always a matrix, and it's important to have in mind we have a complex matrix, because if you target a range of pests, you will also be able to detect many other organisms. That's the primary nature of high throughput sequencing. If you do an RT-PCR or ELISA, you will know you target this virus, I target this species or this race of fungi. But for high throughput sequencing, you know that you may look for this virus, but you are able to find any other virus potentially that is in the matrix. And so think always, my matrix is in fact an ecosystem with many organisms. I show you several examples, and you will be able to detect this organism, even the ones that have nothing to do with the symptoms, for example, that you have observed, or the health of the plants. On the other hand, and in rare case, you will work from isolated organisms. For example, you may sequence pure culture on a plate. You have isolated the bacteria, you just sequence its genome in order to see if there is some pathogenicity related genes, if you have some interesting genes on the genome. But even if you have a single insect, you can sequence it, you will also generate sequence from bacteria. And if you sequence, for example, the RNA from RNA viruses. So have in mind, we always work with complex sample, and the high throughput sequencing is able to detect potentially any living organism in a sample. There are restrictions, because depending on the methodology, you are able to see an organism or not. But think about, we have a complex matrix anyway, which can complexify the interpretation of the results. So that's for the sampling. So same as before, but keep in mind, it's always a complex mix of organisms. Okay, let's have a look on the second step, nucleic acid extraction. For nucleic acid extraction, and whatever your topic, you might be a virologist, a bacteriologist, a mycologist, nematologist, whatever, there is a huge diversity of protocols used. If you have a look on the literature, you can start from DNA, the genome. I want to sequence the genome of my pathogenic bacteria. I want to evaluate the spores, the species of fungi that are in my spore trap. I start with DNA. I may start also from RNA or small RNA once I'm interested in viruses, for example, because if I work on DNA, I will never look and never find the RNA viruses. Double-stranded RNA is also another way to check and detect and target the detection of virus. So what I will ask you to do now, for just a bit less than 5 minutes, look in your most recent publication that you have read on, not THS, but HTS, sorry, whatever it is. It may be yesterday, it may be last week, last month, whatever. Have a look on the most recent publication you have read and look what is the nucleic acid extraction, what is the protocol used. And the key question is there is some specificities for really this single step. So it's easy, you go to material and methods. Usually you have sampling and after nucleic acid extraction. So have a look there. We will, I want you to have a look. And once you have that, please send it in the chat your observation. Is there particularities or not? And it's important that once you have it, feel free to really chat and put it on the chat, because the interest of being so many together is that we can have variety of view and a variety of constatation and of observation. So let's go for that. I'll let you around 5 minutes. Please look on the last publication you have seen on high throughput sequencing, and well, not a review, but actual publication with work in the laboratory. What was the extraction protocol for nucleic acids? Was it something specific or not, or they used the kit as always?
So Sébastien, just to tell you that the participants, they cannot share their responses in the chat. So what I will do is that I will copy the answers myself in the chat.
Okay, great.
So yes, we only got 3 answers so far.
Yeah. Yeah, there's already interesting comments there. 3 but of quality already. Okay, so I clap again to catch your attention somehow. Just what we had here in the chat, there were in fact several responses, and that's really nice. How can I enlarge the chat myself? Okay, let's do it. So what were the observation? In fact, we had commercial kits. We have mobile kits and other commercial kits. There are different ways, yes. Using CTAB, for example, triazole methods, Keygen RNA kits, again mobile, QIAMP virus kits, triazole RNA. I wanted also to share with you, for example, one publication we made. It was already a few years ago on pistachio. So, like not a common matrix. The extraction was silica purification and lithium chloride precipitation. What's the key message that we can get from this? Is that in fact these are similar or identical extraction than for classical RT-PCR. There is one interesting point related to it will depend on what we want to sequence. If we use SMRT, so the one of the technology, or Illumina, another technology. We'll go on the technology after. That's true that some of the new technologies, and we will see it in a few minutes, they can generate very long reads. And so for them, it's advised to really have an extraction which allows to have long nucleic acid, long DNA fragment, in order to achieve and to harvest the advantage of the technology. But globally, what you can have in mind, in fact, is that it's similar to what has been used. We can go to the pool, but I think I have already put. We see the survey. We have a second survey which is possible to carry on here. So I put it here for you. The nucleic acid extraction purification protocol are identical to the used one by RT-PCR. There's sorry. No. Okay. I get a message in red, which is okay here. Okay. There was just a blocking. Sorry. So here are the votes. So you are currently voting quite quickly. Okay, still have 10 seconds to vote. Here it is. There are 40 responses. It's ended. So indeed, you are right. In fact, and as I told before, and I want to underline this again, the protocols are similar. They might not be strictly identical, but mainly the majority they are identical or very similar. So that's the point with high throughput sequencing, sampling, and nucleic acid extraction. There is not that much changes for that.
Sébastien, there is a question in the chat. Do you mean that the high concentration of RNA will deliver data with long sequences?
Sorry, so the question is, could you repeat?
Do you mean that the high concentration of RNA will deliver data with long sequences?
No. The key point we will see afterwards that in terms of quantity, there is. I will answer it afterwards, because now we will have a look on the quality control of the nucleic acid, and we will be able to discuss it more in details for that. But that's a very good question. That's a good question. We'll go back to it. So this is like RT-PCR, but sometimes we might need just a higher quality of the extract. So like it's important and it's really mandatory for before sequencing, there is always a quality control. And that's something we do not have in RT-PCR. I mean, we extract nucleic acid and we make the RT-PCR. Here, so far, quality control is always done before the sequencing. So that's an important point. And for that, we will go for the RNA. And I will first answer here an interesting question, is the quantity. Do I need a big quantity in terms of microgram of RNA to carry on my high throughput sequencing? And this is the same for DNA. But for RNA, when I started using it like a decade ago, we needed a lot of RNA, 5 to 10 microgram. It was a huge amount, in fact, in some cases. In other cases, it's not, but we needed a lot of RNA. Now, you can go to 1 microgram or even less with specific protocol, which means the quantity in microgram is not a limiting factor at all for us. The limiting factor is the quality. And the quality might be important. I will give you some overview of the quality of RNA, how we control it. A technology which is used by nearly all the sequencing facility it is called a bioanalyzer from the company Agilence. So what does it mean exactly? You see here the chip with 16 wheels, and you will put 1 RNA in each of the wheels. There is also a control. And the idea is basically to make a chromatography, to make a migration of your extract depending on the size. It's like making an agarose gel, but much easier and much quicker. Against the one that I've done agarose gel to verify the quality of RNA, we all know that it can be complex and lengthy. Here, you just put on a chip, and there is a capillary migration, and thanks to phosphorescence emission, you can quantify and evaluate the size and the quantity of your RNA extract. This machine can also be used for DNA, but I give here an example on RNA. So what do you get on the electropherogram? You get this kind of observation. You get the marker, you get the profile with the region, fast region, so short time, so it means short RNA. You have a first peak corresponding to 18S or 16S if you are with bacteria, for example. You have a second peak, the long subunit of ribosomal RNA of various size depending on the organism, and after again, nothing. This is what you expect. The RNA has migrated depending on the size, and you get 2 peaks corresponding to the 2 ribosomal RNA units. So, based on several elements, and this is the arrow that you see here, the software with the machine will calculate a score, which is called RIN, Relative Integrity Number. And this relative integrity number will take into account the height of the ribosomal RNA and the height, the quantity of the intermediate RNA. And the best RNA that you may have is this one, intact RNA, 2 nice peaks, nothing else. That's the perfect one. The second one, it's a partially degraded sample. You see the peak of the ribosomal RNA is much smaller. So with the long subunit, it's still very, very small. And you get much higher peak, which means a much higher quantity of RNA here at the very short time, meaning very short fragment. And this is a sign of ribosomal RNA degradation, which means that we could have a risk of, for sure, the mRNA will be degraded, but the viral RNA could be also degraded. And another example here, RIN3, you get this kind of graphs. There is no more ribosomal RNA, but we get a huge amount of very small RNA, meaning that the RNAs have fun. The RNAs has just cut down the RNA. What is important, to have in mind, I will not go into details, but it's really to have an overview of the integrity of the RNA. You can compare your sample together. But what it cannot do and cannot say to you is that my sequencing will work for sure. It's a good indication that your RNA is of good quality, but you're not sure. You can do the sequencing actually. Once you discuss with a sequencing facility, they will tell you, yes, for RNA, I request a RIN of 7 or 8. And in fact, if you used to work with plant and are making this kind of analysis, it's very complicated. Often you get much lower RIN, but it doesn't matter. We will go a bit more on that after. We can have also a graphical representation of the RIN. It goes from 0, I get nothing, with intermediate value. Let's say here 2.6, we have a huge degradation. Here there's still nothing, there's very small ribosomal subunit. And the best one is here. So in order to evaluate if you have well in mind the RIN story, we can go back to the VUCLAP. Again, you can scan the QR code here, and I will open a new question. You have an electropherogram that appeared, and you have now 35 seconds to answer to the fact that looking at the image that you see, if you evaluate that the quality of the electropherogram is very bad to excellent. So,
Sébastien, just to let you know that there was 2 questions and I put them in the chat.
Okay. Maybe what I propose, Charlotte, I will answer this question once I finish the laboratory part, because there is interlink between the different steps. Okay, so it's no more possible to vote. So what you see here, this was this image, and in fact, most of you evaluate as fair, bad, or very good, but indeed, we have something which is fair. Why? That means we still have the ribosomal RNA peak, but we see degradation there. So it's clearly not excellent, nor very well, nor very good, maybe fair, but it depends how we consider it. But it's neither excellent, neither very bad. Second exercise, you have another electropherogram here. How do you evaluate it? This one. Just look at this. Think on it. You also have 35 seconds to respond. Okay, I think we're close to finish. Okay, so let's have a look on the results. Bad or very bad. And indeed, it's not a good RIN here. If you look, you see yet a little bit the ribosomal RNA, but you have a huge amount of RNA, small RNA that has degraded, indicating a strong degradation of the RNA, the ribosomal RNA. And last question here, last electropherogram, how do you evaluate the quality of this one? So we have more than 30 voters. It is rising. 42. Just participate to self-evaluate yourself. Okay, let's have a look. Excellent or very good? Indeed. In fact, we have with an excellent RIN, because we see very well the 2 ribosomal RNA. The only backward is that usually this longer RNA should be higher, but indeed it's a good level. So let's go back here. Indeed, we have fair, excellent, bad, very bad. I wanted to underline one point here that you have to keep in mind if you work with viruses and RNA extraction. And it is also a question in the chat. What is the lowest or the minimal RIN value for a best optimal read length? there is two elements. First, what is the minimal RIN value for detecting a virus? For example, using a short length sequencing like Illumina. There, we had very good result with sample with only 2 of RIN, which means that even if the RNA is degraded, you still have short fragments of the virus, and so you can sequence and detect them. So even if you're the sequencing facility provider, they will tell you the RIN is too low there because they used to have RIN of 7 or 8 because also they are doing transcriptomic with very fragile RNA, messenger RNA. Here, our viral RNA might resist better, and also, anyway, the viral RNA can still be detected. It doesn't mean that you do not have to try to reach the highest quality. A high quality is always better. But think about rare sample. You have no more the ability to re-extract your sample. You may risk the sequencing with lower RIN, at least with Illumina. It can give good results. You can detect viruses. What I don't know if with a low quality I miss viruses, because as I don't detect it, I don't know if they were there or not. But you can still detect viruses with low RIN. That's the first question. But always better to have better RIN. Second question that were there is related to the length. And here, if you want to have, for example, just to sequence full-length virus, in that case, you should have really the best RIN possible. Because if you have a low RIN, that means that your RNA is cut, has been degraded in small fragments. This means that if you have with 8 kb virus, it will be very difficult to have a complete sequence in a single fragment, in a single DNA, a single RNA, sorry, because your sequence, your RNA has been degraded. So if you look for long fragment, it's better to have a longer RIN. But with short fragment sequencing, that's okay with that. And that's the same also if you look for bacteria. You want to sequence the genome of bacteria. The length of your DNA, the longest fragment you can have with long fragment technologies. So also it really depends on the application that you want to do afterwards, but we will review the technologies after. Okay, so that's for the nucleic acid extraction and, importantly, the quality control of this nucleic acid, which is a really important step. Library preparation. What does it mean, library preparation? It's in fact to make the form of nucleic acid which is compatible with the sequencing platform in sufficient quantity of the appropriate size. All the words are important in this sentence. I took care of that. That means that you will prepare your extracts so it can be sequenced on a specific platform, which means that if you use Illumina, there is a certain library preparation. There are 10, in fact. If you use IonTorrent, this is another one. SMRT Pacific Bioscience, another one. MinION, another one. And you need a sufficient quantity of the appropriate size. For example, for Illumina, the final size of your library is very important in the yield that you can have. So that's the goal of the library preparation. And for that, basically, and we'll go back to this after, is just putting adapters on the both extremities of the extracted nucleic acid. For the library preparation, and we describe them in the guidelines quite in details, we have 2 groups of protocol. What is called amplicon sequencing. This one is PCR based, which means from my nucleic acid, I will just make the PCR using primers, and I will sequence the product. Sorry, I hope you don't have too much background there, just making work outside. But that's the amplicon sequencing. And this is done, for example, for microbiome studies. If you want to study the bacterial or fungal population, you will design primers that amplify a specific region, 16S for bacteria, and part of the 16S, not the whole currently, or ITS, the internal transcribed spacer, in fungi, for example, or COI for insects, whatever. You will amplify like a barcode and sequencing. So you generate a PCR product and you sequence it. Shotgun sequencing is the second group of protocol. There, instead of targeting a specific region, what you do is just you take all the RNA or all the DNA that you have and you cut it in small pieces. And these small pieces after will be adapters will be added to these small pieces, but there is not this PCR targeting a single region of the genome. For amplicon sequencing, you can also read, for example, gene targeted, and for shotgun sequencing, you can hear about genome sequencing. We take shotgun, we take the whole nucleic acid, we share in small pieces. That's it. So the key message on library preparation, it's a long process. This is often quite a long process, but it's only about classical molecular biology. You share your DNA, you digest it, you ligate it. I mean, ligation of nucleic acid. You make amplification. It's really the basic steps of molecular biology. Nothing really particular there. To give you an example of the protocol that can be used for viruses, again, there will be other examples on insects or bacteria later on. We have, for example, we can start from vector, from plants, or for whatever the source. We know that inside of it, we can have very different viral sequences, either DNA viruses, RNA viruses, viroid, or even viral sequences integrated in the host genome. And in fact, what we had this in this review and opinion paper published last year, we compare, and that was already done before, different extraction and library preparation protocol. And with a graphical view, you can see that, for example, total RNA, we can detect specific kind of viruses, nearly all of them. That's also the case for small RNA, although more difficult for the integrated viral sequence. If you use double-stranded RNA, we can detect quite a lot of viruses, more difficulties for the DNA viruses, in fact, but not the integrated one. And VANA, which is a purification of viral particles by ultracentrifugation before sequencing, you get only some of them. I don't go into details, but the key message here is that your protocol for library preparation, you have to think about what is the consequences on what I can observe. If I want to analyze the bacterial population of a sample, I really need to care about the primers that I will use, to be sure my primers will generate and will be able to amplify the sequence from the bacteria that are present there, and that my amplicon, my amplification product, will be representative of the actual population that I have in my sample. So that's also an important step. Library preparation do impact the results. As an example here, it's one of the protocol for Illumina from RNA. You get your poly-A RNA. If here there is oligo-DT purification, but for viruses, I really do ask you to avoid making mRNA purification. You will lose plenty of viral sequence doing it. The non-poly-A viruses, for example. You share it in small pieces. You reconstruct the first strand and second strand cDNA, really classical molecular biology. You made an end repair, you had A tailing. You see here this protruding A adenine base. And so just classical molecular biology. And after you make a ligation with specific adapters, and you get your final product here, where you have your DNA, the DNA of your sample, and specific adapters. I will go on the adapters just after. Have in mind that there is plenty of kits. For example, Illumina here, there is at least 11 kits existing and hundreds of different protocols, either to analyze your RNA or your DNA. But among all these protocols, if you look some elements, if you look the recent publication, you will find quickly what are the most popular one, whatever the best with which you're working. So just if you analyze the bacterial, fungal, of insect composition, you will target either a 16S RDNA for the bacteria, the HTS from the fungi, and the cytochrome oxidase, for example, for the insect, which will allow you after to assign a taxonomy taxonomical level to these sequences. So that's for the library preparation related to either amplicon or shotgun based. Last step of the laboratory part, this is the sequencing itself. And there is 4 available sequencing technologies: Illumina, Ion Torrent, Pacific Bioscience, and Oxford Nanopore technologies. Think also and have in mind that there is symmetry of technology. The Roche Ethelix, the Helicos, the Pollinator, the Complete Genomics, the Solid are all technologies that have been developed during this last decade, but that failed to deliver, to compete with the surviving ones. For all the sequencing technologies, and I show here an example with the 4 main current sequencing technologies, specific protocol of library preparation have been developed. The library preparation is really linked to the sequencing technology and sequencing platform. They are not interoperable. And somehow they are all based on the classical library preparation. It's about shearing, repairing, ligating, amplifying. Classical molecular biology, but a more complex and a more longer protocol. So there was the possibility to share with you 4 videos, but I just put it here, and we will send you a link toward this video if you want to have an overview of the 4 main current technologies. And in fact, the idea is to compare a little bit this technology based on the detection method used, the nucleotides used, the enzyme location, amplification, and so on. We will not go into details there, but you will receive the link toward this video to have a look on the available technologies. Be aware that currently Illumina has like nearly monopoly. I don't know the percentage, but I would guess like 90% of the market is used by Illumina technology, a little bit more for torrents, while Pacific Bioscience has a really specific niche of long reads, and Oxford Nanopore, the MinION, is like the super fancy one, but not yet so handy, so easy to use, and with yet some limitation in terms of accuracy. Anyway, I have for your information here put a table with like the comparison of them. You will see the detection method is either by fluorescence or electric signal. The nucleotide, they are pushed into the flow cell through a mix or one by one. The enzyme has some specificities, the amplification too. The length is important because you see there is the 2 short length sequencing and the 2 long length sequencing, PacBio and MinION. They have applications that are interesting there also to reconstruct genome and so on. And just to show you, we'll try to what is a sequencing flow cell. In fact, it's as simple as that. So have a look. I try to put it well. Yeah, this is Illumina sequencing flow cell. So just like a cell from microscope. In fact, here is just in each of, you can see here, in each of the well. So you have the line in each of the line. You can have millions of sequence generated, even billions now. So it's quite simple. You will put and inject, the machine will inject the library into this simple flow cell, and afterwards there will be a progressive sequencing with different technologies, different methodologies, depending on the technology. But have a look on that. I will share with you the link to the video. So if you are interested in digging a bit more in the sequencing technology, feel free to go. Just have a look on a few years ago. I give here some examples that are now more and more outdated, I must admit. It's just to underline that within a single sequencing technology, Illumina, you have plenty of machine. And we will have a look there on Monday, once we analyze, well, if I want to use a technology, what How can I design my laboratory? Which tool do I need? Would I need to outsource or not? Yeah, we will address it on Monday. The frequency for use on virus discovery or characterization, it's mainly Illumina. The frequency of use for sequencing spore traps, it's Illumina. It's really the key players. There are no progressively more application related to Oxford nanopore, but this is still rare, I would say. But they might, if the technology continue to evolve, that might grow. What I wanted to explain with you to go a bit more in details is the point of adapter. Because I mentioned to you there are adapters, but you can ask yourself, what are these adapters? What do they mean? Have a look here on the sequence. It's a sequence, a library. The library is ready. You see we have to insert sequence. This is what? This is the sequence that has been amplified here because you see by a forward and a reverse primer. So pretty easy. You have generated a PCR to get the insert sequence. And for example, you will sequence this COI to analyze the insect population there. If you do the amplicon sequencing, what is there with amplicon sequencing? You have the first PCR. You remember, amplicon sequencing, it's a PCR. And this PCR is in fact using composite primers. You have the forward primers, which can be a 16S primer targeting a specific region of the rDNA. You have your reverse primer, which is also targeting a specific region of my 16S rDNA, 300-400 bases apart from the other one. What is important is that this is, I call them composite primer, because you will order a primer that in fact in the 5' you have what is called sequencing primer bind, and on the 3' you have the primer, which means that your PCR product will in fact already be a sequence from your target plus already an adapter corresponding to sequencing primer. And what is done for amplicon sequencing? There is a second PCR, what is carried out, and this time we use another primer, which is also specific. And this primer, it contains the sequence of the sequencing primer to be able to bind with the PCR product of the first one. You get what is I5 or I7. This is a tag. And this is something very important. We'll discuss it next week. The tag is a sequence of around 8 nucleotides of a specific combination. And in fact, for each of your sample, you will use different combinations. And after, you will put all your samples together in a mix, and you will send your sample in the line of the flow cell. And in fact, afterwards, you generate the sequences, and thanks to these tags, these I5, I7, you will be able to retrieve and to say this sequence belongs to sample 1. This sequence belongs to sample 2, to sample 3. So you will be able to demultiplex, to sort your sequence per sample. And so at the end, you end up with a composite DNA. And the P, in fact, P7 and P5 parts are in fact to bind your DNA to the flow cell, because this flow cell, you have billions of nucleic acid that has been attached to the surface. And thanks to these nucleic acids attached to the surface, they are complementary to the P5 and P7 sequences. So you can link your DNA to the flow cell for the sequencing. So that's what means adapter. So it's about adding a sequencing primer. It's useful for starting sequencing on Illumina technology, adding a tag, and adding a sequence allowing to fix the sequence on the flow cell. If you want to look the shotgun sequencing, in that case, you see the insert sequence, we do not have the primer anymore. It has been the whole DNA, the whole RNA that has been cut in small pieces. And remember this A overhang, T overhang that link together will ligate, in fact, already a composite oligo corresponding to the sequencing primer, an index, and the sequence allowing to bind to the flow cell. And there is, in fact, a ligation of this adapter that is carried out. So you see a bit the difference in both, but at the end, this is the same kind of things. Flow cell, binding, tag, and sequencing oligo, and after my DNA, the DNA that I want to sequence. Okay, that's for the laboratory protocol. Maybe I can have a look if there are some specific questions. So I can have a look. What is the purpose of having long RNA fragment for RNA sequencing if then it is fragmented in short fragment? That's why, in fact, this is not so. It's not so important to have a very good quality. But what you never know, and I have no proof of that, but that's my educated guess, is that the speed of degradation is different between RNA sequences. And it goes back to another question that is here. Will RNA from a capsidated virus be more resistant for degradation? Yes, clearly. That may be also an origin of the fact that even with very low RIN, we can reconstruct the whole genome of viruses. So indeed, I do not understand the question for long RNA sequencing. If we have quality 80% of long fragment, if it can affect our results, maybe if you can rephrase it, it would be nice. Yeah, so the question is it possible to sequence a degraded RNA? In fact, yes, because even if you have long RNA, you need to shear it in small pieces before sequencing it. So it will be sheared, but it is sheared on a very controlled way. I mean, the intensity of the protocol is adjusted by the person preparing the library in order to target a certain length of the fragments. And for Illumina, the best is around 350, 400 nucleotide on average, more or less. It allows you to have the highest yield on your flow cell. If you put on your flow cell here too long fragments, they will not attach well to the binding oligo. And so it's not so efficient for that. But yeah, you need to at the end have a specific size. But the good point, and we will see it just after this point 3, is that even if you have short fragments, we will assemble them together. And so we can, even with very short fragments, reconstruct a full genome. For example, I mentioned one protocol of small RNA sequencing. That means that you will sequence a sample with length of 21, 24, 25 nucleotides. And despite this, you can reconstruct a full genome of a virus. If you look on the amplification, the amplicon protocol, at least you need, for example, a DNA for which the size is higher than what you want to amplify. If you target a region of 400 nucleotides, it's really advised in that case to have longer fragments. Whether, yes, you could amplify yet fragments because the fragments quality is like a Gaussian distribution. Even in bad sample, you still have long fragments, but much less, and you will create bias. So that's important for that. I think I've answered the different questions on the lab. If you have still other questions related to laboratory steps, we will have anyway a question and answer at the end of the presentation. Let's go for the other points.
Sébastien, just to let you know that we have 25 minutes left.
Yeah, but that's okay. Thank you. Bioinformatic protocols. Key point. What's happening now? We have a balance. Until before the high throughput sequencing, this was a lot of lab, a bit of bioinformatics, a bit of informatics. I mean, analyzing the CT, making a bit of statistics. But now with high throughput sequencing, we revert the balance. The informatics is becoming really very important. I put you Harry Potter, and it's an honor for you to have in mind, and we'll go on this next week also. With bioinformatics, you can generate as much false positive and as much false negative as you want. You put a bad parameter, a bad threshold, you put a bad database, you get wrong results. So it's really important. And another key point in the result interpretation, that's why we put a specific point in the guidelines related to result interpretation, is that we often see the life as black and white. Where is the clear cut between a positive result and negative result? And in high throughput sequencing, it's more complicated. We don't have this 50 shades of gray, but 1,000 shades of HTS. Because in fact, you will always have clear results, either clearly positive, clearly negative. And you will have in between a gray zone. Wow, am I sure this is this bacteria? I'm not sure this is really this fungi or this virus. So it's more complicated to interpret at the end. So let's go on the 3 main steps of bioinformatics. The analysis of raw reads. What is the read, in fact? This is the reads have what is called a FASTQ format. This is in fact a line with a name here. Here this is specific correspondence for Illumina. You have the sequence that has been generated by the sequencer, and you get the quality score of each and any sequence. That's important. You generate 100,000 billion sequences in a day. Each one of these sequences has a quality score, and the same. They all have a quality score. Here it's like complex quality score with letters and so on. You can also have quality score with numbers. Here it's between 0 to 40, which is the usual way of having quality score now. But each one of your base has a quality score. And in fact, this quality score is important because you start the analysis of your sequence by eliminating reads of nucleotide that are of bad quality. What importance could it have? It's because a bad quality can be a wrong base. So it can introduce you wrong mutation in your sequence and can so puzzle your interpretation. Bad sequence can be also like random bad sequencing, and can introduce in your analysis a chimera. So it might be important to make a very strong quality control. For that, let's have a look on the next pool. This is one question that I share with you here. So you can go, I will also put here the QR code so you can go on there. What is the importance for you of nucleotide accuracy, and so the strength of the quality control once you want to detect a virus in a sample by RNA sequencing? What do you think? Is it essential, important, interesting, really no matter? Good, we have like 34, 38 questions, so we have a look on what you have. So the majority of you say it's essential, some say important, interesting, and really no matter, very few. And there I will maybe astonish you, because in fact it's not so important. It's between important and interesting. It's good, the nucleic acid accuracy when detecting a virus, but not so much, because you need a lot of errors in order to misassign your virus. You need, for example, if you see the species demarcation criteria, which is 20% of mismatches or 80% of identity, you need very bad sequence to really puzzle it. In some cases, it's important. It's always good to have high quality. But if you get, for example, in a sample, you don't detect viruses, or you have received a data set and you have, unfortunately, very low number of sequence of high quality, it's worth testing again with lower stringency of the quality control. So you can have a look if there are also viruses there. So it's important, but not so much. Other question. What is the importance of nucleic acid accuracy when analyzing microbial communities through amplicon sequencing of 16S rDNA? What do you think? Essential, important, interesting, or it doesn't matter? So I have like 30 responses. Let's have a look. Right. Let's have a look here. Essential. And here you're right. Here it's really essential, the accuracy. The quality control needs to be very strong once you do amplicon sequencing. Why? Because here, 1, 2, 3 mutations can change completely the taxonomic assignation of your read. So here, it's much more important. So the take-home message there is that the strength of your quality control will depend on your application. For some application, it needs to be very, very stringent. For other, you can be more relaxed. Okay. So the raw reads, if you look at this, we have the raw data with index. You can just use the index to assign per sample, sample A, B, C, thanks to the small index. Remember this I5, I7. And you still have this kind of FASTQ file after you make the quality control. This means that some reads will be eliminated or some part of the reads will be eliminated if they don't reach the quality control. Other way to analyze raw reads, you can remove redundant reads, which are reads that are 100% identical to another one. I don't do it if you do 16S or amplicon sequencing, whatever it is, but it might be interesting with shotgun sequencing. If you use paired reads, the Illumina technology, which allow you to sequence your sequence in both directions, you can pair them or merge them. It may improve the assembly later on. But that's the 4 main analyses that you can do with raw reads. Let's have a look on the 6th element, which is the identification of the target. The first step of it, when you want to identify targets, it's to go from the individual reads to contig. And what's a contig? It's a set of overlapping DNA segments of reads that together represent a consensus region of DNA. We have 2 options. We can assemble versus a reference genome. I know, for example, I have Ralstonia in my sample, and all my reads will be compared to my reference genome. And I could generate 1 contig, and you can see here, I put contig together because they have homologies. And I generate a longer sequence. And that's pretty interesting to have longer sequence because after it's easier to annotate or it's easier to analyze the whole genome. So that's an important way to do is to put together the reads into longer segments. You can do it by comparing with a reference genome with the fact that you will have regions that are not sequenced. And here it may be due to the fact that your reference sequence is from a strain that is different from your strain, the strain you have sequenced. Some region did not exist. Some region of the reference genome do not exist in your sample. And at the opposite, this approach, you may miss some region because, for example, at this place in your genome, you have another sequence completely different, but which can be important in order to evaluate your bacteria. That's why there is another option, which is the de novo assembly. For the de novo assembly here, you will put all the reads together without any guiding reference. And you can again generate the different contigs. But the interest is that in fact, you can fill the gaps because you will assemble the puzzle, assemble the contigs related to your sequence and not a reference genome. It gives you that. Here are all individual reads with their name, and they have been aligned together. Each blue indication is a mutation compared to the consensus. But here is this kind of, you see, on a single position, you have hundreds, sometimes thousands of reads. So the de novo assembly, it reflects the real genomes in the sample. That's a positive point, but you need more computing power because you will compare millions of reads with millions of reads. Whereas mapping of the genome, it's quicker because you analyze all your reads against single reference genome, but there is bias between strains, isolate cultivars, and the reference sequence. Nevertheless, there is no opposition, and some bioinformatic pipeline use both to reconstruct context. Amplicon sequencing, the story is different because here you need to group the reads together. And for example, we have 4 bacterial species in a mixed population. You can see 4 colors for these species. We amplify them, a specific region, we sequence this, and we generate, as it is here, plenty of bases of reads from sample A, from sample B, from sample C. Stringent quality control, and after what is done, you assemble the sequence to each other depending on their identity. You define, in fact, a certain level of identity, usually 97 or 99% of identity, and you just group the sequence together. You will not build contig because you have amplified a single region. And you, in fact, determine, oh, this is cluster 1, cluster 2, cluster 3, cluster 4, and so on. Based on that, well, and just to mention, there is a third way. So first way, we assemble the reads, overlapping reads to construct long contig shotgun. For amplicon, you group the reads together, depending on the similarity. And a third approach that can be used in both cases is analyzing each individual read. In fact, it did not exist a few years ago because it was too demanding in computing power, but now, with the improvement in computing power, with the algorithm optimization, we can do it. I don't go into detail, but there are several software that allows to make analysis of each individual reads. The identification of target after is related to a comparison with database. For that, in fact, there is like the trinity, which is important. So either you have with your group of sequence, you have with your contig, you will compare them with the database. And keep in mind that the database that you use, the algorithm that you will use to compare both, and the parameters used for the algorithm, the threshold and so on, these 3 elements will completely influence the results. And it is very important to be able to evaluate what is the best database, what is the best algorithm, what is the best parameter to make this comparison. For that, there is the most popular one is the BLAST algorithm, which is really the key, well, I mean, the most popular one, at least for shotgun here. It's another algorithm for amplicon, but BLAST, in fact, it will scan a database for words of determined length and calculate a threshold. After it, well, I will go directly into the example here. What's happening with a BLAST algorithm? Here is the sequence generated, either a single read or a contig. The BLAST algorithm will cut the sequence into words of 5 letters here, for example. And you see there is all these words of 5 letters and the different elements, which means that from my sequence, I have 40 different words of 5 letters. Can be nucleic acid here, but also amino acid. By default, for example, if you use on NCBI, it's 28 letters. But here, we make the example of 5 letters. What will it do now? It will use all these 40 words of 5 letters and compare it with the database. And it will do the same with the database. It will cut sequences into small pieces of 5 letters and will look for 100% homology. It's really quick to do. Is it completely identical? Yes, no. And in fact, for example, it found a hit. Okay, I get a hit there. First point, good. 100% identical. What will do the BLAST? The BLAST will in fact encore the 2 sequences with this 100% hit and progressively will analyze in one direction what are the homologies, and for each homology or not, it will calculate like a score. And you can see that there are several bases that are homologous and which allow to calculate a score. On the other hand, it will do it on the other way. Here, what you can see, there is one homology, but after no homology. Oh, damn, nothing is similar. What's happening? Do I have a very bad score? In fact, no, because the BLAST is also taking into account small insertion of deletion. And that means he also calculates that, in fact, if I consider this base A as an insertion, I get a very nice homology later on, which improves the score. And the score, this is what I mean by the importance of the parameter used. What would be the minimal score that you will take into account for your analysis? So the blast will stop when the score is going under its own threshold. And after you will see, okay, this threshold is fine for me or not. And I don't go into details, but a key parameter that is used is the e-value. You have here for information, it's not so important at this stage. It's important if you want to dig into high throughput sequencing, but you have different kind of blast program. You can compare nucleotide versus nucleotide. You can compare translated nucleotide versus protein, or translated nucleotide versus translated nucleotide. Each one has its own advantages and drawback. Have a look also is that for each contig you have hundreds of blast hits, but you will see only the first one. So sometimes, it's just a tip, once you make for the first time your analysis of shotgun sequencing data, it's worth to see if your fragments and the first blast hits that you see on your table, if it is confirmed by the second and third blast hits, because the problem with database is that you can have misassignation in database. You can have garbage in fact. So always look the tree hiding the forest. Blast is currently the longest step. It takes a lot of computing power. You need a lot of resource, and it's like the bottleneck of the analysis. That's why a lot of new algorithms are developed to speed it up. Let's go to the last part, the analysis of control. We will let it for next week. Last part, which is confirmation, interpretation, and reporting. This is detailed in the guideline, but I wanted just to underline the problem that we are facing as biologists. As virologists, we have identified hundreds of new viruses, but we are not able to cope with the biological characterization. So there, the guideline, we think also about that in fact you discover a new virus, or you detect a virus that has been recently discovered, you can try to go a bit further if you have resources to characterize it. And we have proposed in this publication, this common publication 3 years ago, a framework which is here. So the goal of today is not to go into details here, but have in mind that the interpretation of the data is really a key. It's really important. So, conclusion of today, this introduction. If you want to put high throughput sequencing, to go for high throughput sequencing, you need to carefully review all these steps, because all of these steps will have an impact on the results. And you have to look at both for laboratory, and you used to do it, but also it's new on bioinformatics. There is also a huge diversity of protocol. The devil is in details, but indeed, you will see quite variable protocols. So an in-depth bibliographical research is important. And keep in mind that you can create bias at any steps, including the bioinformatics. I will show you an example next week. The standardization of protocol. Progressively, we'll have more and more standard protocol, recommended protocol. You will have like, okay, I will follow it. The library preparation, there will be also more easier and easier, or sometimes you will outsource it. I warn you about acute kititis. That's a disease that we detect on the young students. The acute kititis is just following the protocol of a kit, without asking yourself any question. You must understand what's in the kit, the steps of the kit, because it can be important if you have a trouble to troubleshoot these difficulties. So importantly, the kititis is just using a kit without understanding nothing. Don't fall into that. You use a kit. It's nice because it facilitates your life compared to former protocol. But you must understand very well this kit for nucleic acid extraction, but also for the library preparation. What are the different steps? What is important in these steps? So careful review is really important. For the bioinformatics, it was like very difficult a few years ago. You need to have informatics skills, but now you have plenty of Windows-friendly resources that are available. In addition, you have also a web page. You have, I would say, all-in-one software that are available to make your data analysis. Here's an example for viruses and viroids. But if you're interested in bacteria or in fungi, think about the SHIME or the Mother interface. It's like all-in-one interface that requires low amounts of informatics skills. An example of protocol of standardization, and I just want to finish it by warning you with another disease that might expand among the scientists and biologists is the acute click-eyes. In that case, you use a user-friendly protocol of bioinformatics, and you put okay, let's go the analysis. You must understand the different steps that are in your process in order to deliver and to understand your result and to avoid creating bias. So that's what I wanted to share with you today. Thank you for your attention for this. I will stop sharing my screen. And yes, we still have a little bit of time to respond to the question that you may have. I put the question here.
So you had two previous questions. Which are your suggestions for avoiding RNA degradation?
That's for this is really the classical way of working with the RNA. I mean, having a dedicated bench, dedicated pipettes, using RNase-free tips, using RNase DAP, favoring the conservation at -80 with flash freezing and flash thawing also. The lengthier you're freezing or thawing, the higher the risk of having degradation. And after that, it's really based on the plant. I don't know on which plant you are working, but you know better than me what can be the most appropriate kit for your plant. Here in the lab, we are working on several plants, and I don't use the same kit for banana than for cereals than for potato, for example. So there's really classical way of keeping RNA, and again, high quality is really nice to have, and we should target it. But if you cannot have it, but you need to sequence the sample, the risk might be taken. I don't guarantee that you will have nothing. Don't misinterpret me. But you may have good result with lower read, even if the risk of failing is higher. So it's a balance of risk and scarcity of the material.
And then there was a question about the sequencing primer, how it is composed so that it binds every sequence.
Okay, so here it's the only, well, when you use this primer, you will design yourself the 3' end of this composite primer, which is depending on your targeted region. You want to sequence ribosomal DNA from bacteria, HTS from fungi, COI from for insects, whatever. You will design your primer there. And so this primer will give the specificity of the amplification. What's happening is that in 5', you can amplify fragment with a 5' overhang. As far as the 3' is really bind to the target, and that's what you aim during all the PCR, this level can, this 5' pen is maybe not important in the first step. Afterwards, it will be included in your PCR segments. And this sequence, in fact, there is, well, for Illumina, there is recommendation. The sequencing primer must be of this sequence. It's really important to have exactly the same sequence as recommended by Illumina. And that's the same for any library going to the Illumina sequencing machine from years. This is the sequencing primer, and it must be always the same. And after that, you know that this end of your PCR product is this sequencing primer. Whatever the target region that you have in between, you have the sequencing primer. Once you go to the second step, adding the tag and the binding oligo, again, the binding oligo is the same as, not ever, I don't remember exactly, but it's the same from years. The oligo binded on the Illumina flow cell is the same. So this sequence is also the same. What's changed is the ID, the tag that you put. And at the end of this other oligo, you have a complementary sequence which is complementary to your sequencing primer. So it allows you to get this DNA ready to sequence with your target sequence in between and adapters at both extremities. Again, for example, in Minion, the protocol is different. It's not strictly the same. But for Illumina, you always need to have these two kind of adapters on both ends.
So I think we already answered, but there was a question, if you got a RIN of 2, 3, is it enough to go on, or?
Yeah, I mean, it's like I said, you have a higher risk of having a not so well sequencing. But at the end, on my personal experience, that we get good results with RIN of 2, 3. But that doesn't mean that for any sample in any situation, we will get a good result with a low RIN. So it's a balance there between what's really possible. Do you see a way to improve your extraction protocol or not? I mean, if you test it's the 5th kit that you test and you cannot give a higher RIN, it's worth trying and taking the risk. It's always a higher risk. But if you know that, for example, other publications are using, or your neighbor are using another kit that seems to work better, it might be good to use it. So again, there is no clear cut, like I explained, this 1,000 shades of grays. I remember having taken this risk for several samples because there was no way to have again a sample and we detected easily viruses in there and we get the whole genome in there. It doesn't mean it will be the case in each situation.
And there is a last question. Can you please advise a DNA extraction method suitable to bulk extract insect DNA from traps for HTS?
I cannot advise a method, but I can advise you a review publication by Piper et al. Or can I don't know if I can find it very quickly. No, I will send the link through Charlotte. I will send you the link of the full video and of this publication because this is a group of Australian researchers. They really review in details all the steps for insect amplicon sequencing as a surveillance tool. They envision it as a surveillance tool, either upon entry, in post-entry control, or even to evaluate the epidemic evolution of indigenous insects. So I will share with you the link toward an interesting publication in which they review each step of the protocol for insects.
Okay, so we have passed the time. So I would like to thank you, Sébastien, for the nice presentation, and thanks all the attendees for being here today. So there will be a satisfactory survey at the end of the webinar or in the follow-up email, and it's really short. Please do not hesitate to provide feedback. And as I said, the presentation and the link to the recording will be put on the website. Our next webinar of the series is on Monday at 2 p.m. and we will discuss about how to prepare the laboratory to conduct a HTS test. So thank you very much, everybody, and have a nice end of afternoon. See you on Monday. Bye-bye.
Bye-bye. Thank you, everybody.